Good morning, everyone. This is Ben, and today I'm going to talk about pollinator guilds and pollen dispersal in two sympatric species of Texas Nolina. So, a brief overview before we start. I'm going to start off with the genus Nolina, get into its pollination biology and pollination syndromes, talk about pollen dispersal in genetic neighborhoods, the consequences of phenological division within Nolina, and get into my study, which is a comparison between Nolina rumpens and Nolina texana, using a floral visitation survey and a pollen dispersal study. So the genus Nolina is a group of xerophytic, meaning dry-loving, polycarpic, flowering multiple times in its life, evergreen plants indigenous to Central America, Mexico, and the southern United States. It is an asparagacean plant, which is a huge family, a cosmopolitan family, including things like onions and the more recognizable desert plants like yuccas and agaves. The genus has 30 species and growing. I think 32 is actually the more recent count. But 14 occur in the United States, and 6 of them are in Texas, which makes Texas the most speciose region for Nolina. And they're characterized by, or as a dense cespitose rosette arising from a subterranean branched caudex. So they have this large water storage organ below ground, and then above ground, it basically looks like a large tuft of grass, except when it has flowers. So the inflorescences are large paniculate scapes, sometimes contained within the rosette, other times rising many meters above the rosette. So sometimes you'll see specimens that look like this previous photo, where the panicle is roughly the same size as the plant itself and within the plant, and other times it'll appear much more like an agave. And the inflorescence contains many small diurnal flowers with cream-colored tepals, and they open over a period of time. So the entire inflorescence may be flowering for a month, but the individual flowers are only open a day. And Nolina are dioecious, which means the male and female parts are borne on separate individuals, and it's entomophilous, meaning it requires insect pollination, and it offers abundant nectar and proteiniferous pollen as a reward. So pollination syndromes are kind of the classical way of looking at flowers and making assumptions, if you will, about who is pollinating. But pollination syndromes are suites of floral traits associated with the attraction and utilization of a specific group of animal pollinators. And there's been a lot of studies that validate pollination syndromes, but some that also urge caution. So it's definitely best to get an actual observation as opposed to just going with the pollination syndromes. And so in 1911, William Trelease, who is a very notable botanist who described 3,000 species of plants, he remarked that Nolina flowers were likely pollinated by insects in the Hymenopteran and Dipteran order based on the pollination syndromes. Hymenopteran are bees and wasps and ants, and dipterans are the flies. And he saw all of the really small flowers on the inflorescence with nectar and pollen, and those are things that typically align with bees, flies, and beetles as well to a lesser degree, but I would say it's more like a generalist flower complex. It certainly attracts a lot of visitors. But so a pollinator guild has only been established for one population of one species, Nolina microcarpa, in northwest Arizona. And this study found that the majority of visitors were Hymenopteran and Coleopteran, which Coleoptera is the beetles. So more research is certainly needed to understand the pollination biology. In terms of the actual pollination process, pollen has to move, and pollen, of course, is the genetic material, the male genetic material, has to move to the stigma, which receives the pollen, and eventually the pollen makes its way to the ovaries if everything works as planned. So entomophilous plants utilize insects to do the actual moving of the pollen. So in order to successfully be pollinated, they have to first attract pollinators. But each pollinator offers a varying degree of benefits. For example, some are more effective at transporting pollen, and this is largely dependent on what the species of plant is. There's no true rule for this. And others disperse pollen much further distances, resulting in a larger genetic neighborhood, which we're going to get into in the next slide. But a couple generalizations here. Dipteran and hymenopteran pollinators travel relatively short different distances between conspecifics, which are species of the same. And Lepidopteran, which are the butterflies and moths, move pollen significantly farther distances. Oops, computer is going a little slow. Okay, sorry about that. So genetic neighborhoods. It's a spread of individuals in a population where genetic differentiation occurs. You could basically think of this like a group of plants that are interchanging genes, or exchanging genes, I should say. And compared to large genetic neighborhoods, small genetic neighborhoods have higher genetic drift. And basically this means either the loss or accumulation of alleles, for better or for worse. So there's a degree of risk involved there, and it can lead to a loss of genetic diversity, potentially imperiling species. So it's certainly in the plant's best interest to have a larger genetic neighborhood. Within Nolina, in Texas, there are some species that flower in late winter and some in late spring. And those actual windows vary quite a bit due to precipitation and temperature, but those are the two main blocks. So this study intends to explore the consequences of floral phenological separation in Texas Nolina, addressing the following questions. 1. How do flowering periods within a genus affect floral guilds? And 2, is there a season-dependent genetic advantage? So are the ones that are flowering in late winter going to have a larger genetic neighborhood, or are the ones in late spring going to have a larger genetic neighborhood? Is there no difference? That's kind of what this study is hoping to explore. And we're frozen again. Here we go. So the two species, Nolina erumpens and Nolina texana, they're very similar plants. They both have inflorescences contained within the rosette. Nolina erumpens, however, is quite a bit larger, often 6 to 7 feet in height and diameter, and Nolina texana is more commonly 2 to 3. But again, they have the same style of inflorescence. Nolina erumpens, of course, produces a proportionally larger inflorescence as well. But the main difference is the separation in flowering periods. Nolina texana flowers from February to May, and really just a month out of that block of time, like I mentioned earlier. But this is the early flowering block. And here's a photo of it. And again, this is the smaller one as well. So this is probably a 2-foot individual. And Nolina erumpens, which flowers in the second block, again, roughly a month, from May through September. And I meant to talk about this, but the distribution of Nolina texana is largely central and western Texas, occurring in the Edwards Plateau as well as the Trans-Pecos. And Nolina erumpens is exclusively in the Trans-Pecos and is also much more uncommon. So my hypothesis is, or I should recapitulate this real quickly, but the study hopes to investigate the consequence of phenological separation within a genus by providing a detailed comparison of the floral guilds and genetic neighborhoods in two sympatric Texas Nolina species. My hypothesis is that the later floral phenology of Nolina erumpens, the larger one, favors a broader array of pollinators because it correlates with the seasonal repopulation of pollinators in Texas. And based on this, I suspect that it will have more frequent visitation from Lepidopteran pollinators. And like I mentioned before, since Lepidopteran pollinators are more effective at dispersing pollen long distances, I expect that the genetic neighborhood for Nolina erumpens will be larger. So now we're going to get into how I'm going to test this. Oh, come on, computer. So in order to create pollinator guilds, I need to sample within the distribution to account for regional variation. So for Nolina texana, that includes the hill country. And so these are the 3 places I'm working with currently. But I haven't entirely decided if these are the ones that are going to make the most sense. For Nolina erumpens, which has a smaller distribution, I'm just going to check out Big Bend National Park and McDonald Observatory. And yeah, this will be determined a little bit later when I have time to go to these sites and explore them. So the first part of the study is to create a floral visitation survey. So this is a visual observation of all of the animals, presumably insects, visiting and feeding from the inflorescences, which I'm going to create 100-meter transects at each location. And they'll be surveyed 3 times during the flowering period, so basically 3 weekends in a row. And each time I do it, there will be 4 blocks that I check: the early morning, mid-morning, afternoon, and evening. And this is to account for all of the various pollinators who would show up. I'm going to photograph the species with a macro lens so that I can identify them later on at some collections. The 2 that I'm intending on using are the Jim V. Richardson Invertebrate Collection at Sul Ross and the University of Texas Entomology Collection. That's how I'm hoping it goes. If necessary, I will also collect specimens to bring there as well. But I'm naively hopeful that photographing them will work. The second part of the study is to determine how far pollen is being moved. And as mentioned before, they are dioecious, so there are separate male and female individuals. And so for each transect that I'm doing a visitation study on, I'm going to select a staminate individual and paint all of the visible pollen grains using a tiny little paintbrush dabbed in fluorescent dye. And this is protocol adapted from a study I read, MAR 2000. And then at the end of the day, I'm going to survey all of the pistillate individuals within a 50-meter radius to look for evidence of pollen transfer. The thinking is that the pollinators will move the pollen, and by determining how far the pollen moves, that should give me a pretty good idea of what the genetic neighborhood is. So for each pollen grain observed, I'm going to record the distance from the staminate individual. Yes. Almost done here, so I'm moving right along. But the statistics I'm going to use are going to be t-tests, which for the floral visitation study, I'm going to use them to determine if there is a significant difference between the number of species or the orders observed. And for the pollen dispersal study, I'm going to use t-tests to determine if there's a difference between the mean, median, and mode of pollen transfer between the species. And again, my hypothesis is that Nolina erumpens, the larger of the species, will have a larger genetic neighborhood because it attracts more lepidopteran pollinators, because it is in the later period of the year when more pollinators are active. So that's my study. Wish I could answer questions for you, but unfortunately I wasn't able to be in class today. But I'd love to hear from you if you do have questions or comments or suggestions, because this is the project that I intend on moving forward with. So let me know. And here is my lit cited. Thanks everybody. Hope you have a good one.